Hi everyone, we are Team 8. Today we will be presenting on 3D bioprinting for artificial liver fabrication. In today's presentation, we will first give some background on liver transplantation, followed by existing challenges with the process. We will then talk about our proposed process of using 3D bioprinting to fabricate artificial livers for transplant, and how it compares with the current process through a technical analysis. Lastly, we will discuss areas of improvement for the future, and then conclusion. Liver, which is the largest internal organ that performs many critical functions, is the second most common type of transplanted organ. The liver transplantation process begins with an evaluation of patients. After having completed specific tests, procedures, and consultations to determine their eligibility, patients are placed on a waiting list. Depending on the severity of their illness, patients may experience a median wait time of 321 days. When a liver becomes available, patients undergo a transplant surgery that can take up to 12 hours long. After surgery, they are in recovery for at least 6 months and need to take immunosuppressive medications for life to minimize rejection. Despite having more than 8,800 liver transplants performed in 2019, there are still more than 11,000 people on the liver transplant waiting list in the US. In addition to the scarcity of available donors, liver transplantation also brings about the issue of immune rejection of the donated tissue by the recipients. As outcomes of current efforts are still suboptimal, scientists are investigating alternative methods such as tissue engineering to provide therapeutic development for liver diseases. 3D bioprinting, a subset of 3D printing or additive manufacturing, offers great potential to fabricate complex tissue constructs from biomaterials with high precision through a layer-by-layer building process. By using patient-derived cells to develop artificial livers, 3D bioprinting is expected to be a promising solution that not only minimizes the immune rejection response of traditional liver transplants, but also provides personalized therapy. As a result, the proposed process needs to produce a functional liver tissue with the liver's many functions, as well as meet the objectives of having negligible difference in cell viability between printed and non-printed cells, and unaffected hepatic characteristics of cells post-printing. Here is an overview of the design process. It is divided into 4 steps, and we will go into details for each of the steps. Prior to the process, induced pluripotent stem cells are first extracted from patients themselves. Then in step 1, a detailed liver image of the patient is obtained through clinical diagnostic technologies such as CT scans and magnetic resonance imaging. The 3D image information is then transformed and segregated to provide instructions to the 3D bioprinter. In step 2, the extracted cells are cultured and differentiated. Falling under human pluripotent stem cells, induced pluripotent stem cells, or iPSCs, and embryonic stem cells, or ESCs, are able to self-renew indefinitely while maintaining their ability to become any required cell types. This property enables large quantities of human pluripotent stem cells to be easily created in vitro for 3D bioprinting. Although earlier researches focused on using ESCs, which are derived from human embryos, the construction of tissue from these cells for implantation in vivo gives rise to ethical issues as well as the need for the patient to take immunosuppressive drugs. However, iPSCs are derived by reprogramming somatic cells with the help of 4 transcription factors. As a result, iPSCs derived from harvested adult cells from the patient are used in this design as they are more compatible to the patient's immune system and suitable for autologous therapies. The iPSCs are first cultured on Geltrex matrix with essential 8 medium in a laboratory incubator set at 36 to 37.5 degrees Celsius. Next, the directed differentiation of iPSCs into hepatocyte-like cells is performed according to the schematic shown on the bottom right, where iPSCs are first cultured in endodermal induction media, then in hepatoblast induction media, and finally in hepatozyme media. The completion of the entire process takes a few weeks. In step 3, the tissue construct is fabricated using a 3D bioprinter. The feeding material of the bioprinter is bioink. They can be composed of cells and biomaterials. The cells are enveloped in a hydrogel, which provides a suitable environment for cell adhesion, proliferation, and differentiation. As tissue scaffolds, hydrogels are composed of natural or synthetic polymers, and each has their own advantages. Currently, there are three major types of 3D bioprinting methods, but extrusion-based is the most common method for printing of artificial livers. It consists of the following components: a 3-axis computer numerical control machine with a microscope, a micro-sized solenoid valve, and nozzles connected to a static pressured bioink reservoir via flexible tubing. Using all of this, the bioprinter provides free movement in XYZ directions, high-resolution nozzle position control, and dispenses cells in very precise volumes. Variables used to control the 3D bioprinting process are the inlet pressure, the internal diameter of nozzles, and the pulse time. RGD-coupled sodium alginate is selected as the biomaterial for this design due to its high biocompatibility with stem cells. The shear thinning behavior of alginate and its properties of non-immunogenicity, low toxicity, and hydrophilicity make it a suitable scaffold that helps to maintain the viability and activity of printed living cells. To further stabilize the printed structure, calcium chloride dihydrate is used as the cross-linking agent. Upon exposure to a calcium ion, the alginate solution undergoes a rapid chemical cross-linking process, and new covalent bonds are formed to stabilize the printed structure. During the printing process, the bioink and the cross-linker are printed through two different nozzles. Factors such as extrusion pressure and nozzle length will be adjusted in advance to ensure high cell viability. The cell-laden bioink will be deposited layer by layer in an array of droplets. Cells that undergo 3D bioprinting are subjected to some level of stress during deposition. Thus, investigations of the viability and biological functions of the printed cells are required using a cell viability test with a fluorescence-activated cell sorting flow cytometer. Moreover, the hepatic characteristic of iPSC-derived hepatocyte-like cells is evaluated by albumin secretion through a human albumin enzyme-linked immunosorbent assay. In terms of expectations, negligible differences should be observed between the viability of printed and non-printed cells. This indicates that the valve-based printing process is gentle enough to print the iPSCs while maintaining their viability. Secondly, the printed cells are expected to be positive for albumin, which demonstrates their hepatic lineage. A limitation to the process is the reduced number of living cells observed post-printing. This is mainly due to the shear force applied on the cells as they are pushed through the nozzle during the printing process. With low cell viability, reduced secretion of albumin will also be observed. As a result, factors such as pressure, nozzle length, and the volume ratio of components in the hydrogel should be optimized. An efficient way to determine the optimum printing conditions is to apply an optimization technique. Parameter optimization index, which is a standardized metric, can be used for our design to evaluate the printed results based on the printing parameters. The index works by simultaneously minimizing the shear stress acting on the bioink and maximizing the printing accuracy. Here is a table describing the impact of printing parameters on our printing objectives. We can see that when the applied pressure increases, so does the shear stress, which causes more damage to our cells, leading to lower accuracy. To balance the printing objectives, two equations on the right can be applied to help us determine the optimal printing conditions. From the relationships in the table and comparing experiments conducted in laboratories, POI values can first be calculated using equation 1. The POI value for a specific bioink can then be normalized relative to the maximum POI that could be achieved in the experimental series using equation 2. The calculated score will be a dimensionless number between 0 and 1. By applying this technique, the optimum printing parameters for our extrusion-based 3D bioprinting process can be determined by finding the highest POI value, where high cell viability and unaltered cell functions are ensured with minimum shear stress and maximum accuracy. As described previously, the supply of transplantable livers has always come short of the total number of required transplantable livers. As a result, the wait time to receive a traditional donor liver continues to increase. However, 3D bioprinting may solve this organ shortage problem by increasing the supply and reducing the associated costs. Currently, the cost of 3D printers and bioprinters have decreased significantly since 2008 as DIY bioprinters designed and created by researchers become more prevalent. Through the advancements of technology and economy of scale, the cost of artificial organs fabricated from 3D bioprinting is expected to decrease over time. Here is a recap of our design process. We first obtain a 3D CAD model of the patient's liver, which will be segregated into 2D slices to provide instructions to the printer. Then iPSCs, taken from patients themselves, are cultured and differentiated into hepatocyte-like cells. Alginate solution, which works as a scaffold for living cells, will be cross-linked with calcium solution, and the resulting bioink is printed through our extrusion-based multi-nozzle 3D bioprinter. Finally, the viability of cells and their hepatic characteristics are determined to evaluate the quality of our artificial liver. By completing the 4 steps, a 3D printed artificial liver is successfully made and will then be transplanted into the patient through a surgery. This concludes our presentation for today. Thank you for listening.